Hello, madams and classmates. We are from Group 5, presenting of the techniques in the field of entomology. I will now pass down to FARHAT explaining of the introduction.
Arthropoda is among the largest animal phylum, consisting of 4 subphylums, which are Crustacea, Hexapoda, Myriapoda, and Cheliceratta. Whereas they have characteristics such as they have jointed appendages, they have chitinous exoskeleton, and also they undergo sexual reproduction. Moving on to objectives. So the two main objectives in this, the first one is to recognize the correct techniques in insect collection and preservation, and also to apply the correct techniques in insect collection and preservation. Moving on to material. The main materials used were styrofoam, nets, containers, collection box, styrofoam cutter, pins, gloves, mothballs, and plasticine, and also tubes. Moving on to the methods. Firstly, the insects were collecting using prepared equipment such as nets, jars, and forceps from various habitats, and also then the insects were transferred into a container, and then they were finally sacrificed using chloroform before being pinned for preservation.
So for the results section, each of us had a good amount of insects caught. So I managed to catch 5 insects, all from the subphylum Hexapoda. So I first caught a strip flea beetle, an orange beetle, a bowfly, an assassin bug, as well as a ladybug. So each of them were very easy to catch.
Okay, so I collected 3 species, which are dragonfly, fine shrimp, and ants, where they all belong to kingdom Animalium and phylum Arthropoda, whereas the ants belong to an order known as Hymenoptera, and dragonfly belongs to an order known as Odonata, and the fine shrimp belongs to an order known as Decapoda. Whereas the ants, they live in nets and can cause allergic reactions, and also the dragonflies inhabit fresh water and they help in controlling mosquitoes, where they are not harmful to human beings. While the fine shrimp, they live in marine waters and serve as bioindicators, whereas the ants undergo complete metamorphosis, and the dragonflies undergoes incomplete metamorphosis, whereas the shrimp undergoes complete metamorphosis.
Moving on, I managed to capture 5 insects, all from subphylum Hexapoda. Firstly is the short-horned grasshopper. Second is the black soldier fly. Third is the Asian weaver ant. Fourthly is the sickle-bearing bush kraken. And lastly is the stingless bee. For discussion, out of 30 insects we collected, 12 of them are Hexapoda and 1 is Crustacea. For life cycle, none of them are complete metamorphosis. For habitat, 11 of them live on land. 1, such as the dragonfly, are freshwater associated species, and 1, which is shrimp, are aquatic habitat. Lastly, 2 of our bug are carrying primary disease, which is the assassin bug carrying the Chagas disease and the bloodfly carrying the Miasis disease. So, in conclusion, through teamwork and preparation, we successfully collected 13 specimens and gained hands-on experience in medical entomology. This project taught us the importance of collaboration, proper specimen handling, and accurate documentation. Overall, it improved our knowledge of taxonomy, medical pointers, and public health, while also strengthening our teamwork and time management skills. Now, lastly, moving on to reflection.
So lastly, as a reflection of this whole journey of the 3 of us collecting insects for our major project, I think we 3 successfully managed to collect a total of 14 specimens in the different locations that we all tried to search for insects. So this include, like, we searched insects in Taman MSU, at Bukit Kiara, as well as nearby parks near our homes. So we made sure we made good use of our time to collect as many insects as we could. Of course, this project involved a lot of time planning, team management, as well as teamwork to ensure that this whole project was done well. Despite everyone's busy schedule, we each managed to cooperate with each other and also coordinate to ensure that we all meet the deadline as well as the requirements of this project. Not only that, I think we 3 have learned that through this project, communication is really important in ensuring the success of this project. So we had to constantly make time to meet each other after class or even during weekends to plan on where to go, how to display our insects, and also share different ideas and thoughts that we all had in order to make our presentation or our insect collection box the best it could be. And I think we all managed to do this all very well and in a very coordinated and well manner. So that's all from us. Thank you.